What's its Padama?
It's Palmer Research.
Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Nonhlanhla Mtshali. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. As you know, we have our WhatsApp line that is open. So if you have any questions regarding the content that is being presented today, feel free to send your questions to 079 454 9535. I will repeat the number again. It is 079 454 9535. So we're going to go into our topic for today, which is paper 2. We are not going to do the whole content of paper 2, but the point of this lesson is to give you pointers as to what is it that you can improve on, what to change from what you wrote on your prelim exams, and hopefully it's going to prepare you for your end of the year exams. So we are going to get on into it. With the content weightings, we have been saying this so many times. So our paper 2, the topics for paper 2 are DNA, code of life, meiosis, genetics and inheritance, and we also have evolution. And as you can see there on the weightings and the marks, we can see that evolution carries more marks, and we have genetics, which also has about 48 marks, and then we have our DNA with 27 marks, and meiosis, which is 21 marks. This information, you find it on your exam guideline for 2021. And then the resources that you need, or you will need when you study, when you prepare for your exams. Remember, our DBE study guides that were shared to you, you got them from your teachers. These are electronic copies of the study guides, and you have our SIP material. We've got our past exam question papers. We have our trial exams from different provinces. We have shared all the 2022 ones from different provinces. You have your teacher notes, the activities that you did throughout the year in the class. You have your exam guideline, your textbooks, the mind, the gap, it's very important. And lastly, we have our website, the www.education.gov.za. And the most important one is this one, www.pocketschool.co.za. Here you are going to find all the lessons that we did from beginning of the year until now. So whichever topic that you are studying and you really don't remember what the teacher told you in class, you can always go to this website, to our YouTube channel, and you will get all the topics, and you can go back and listen to those lessons again. So I'm going to start with the DNA code of life. With the DNA code of life, we've been told so many times that what is important here is that you must know all the terms. So terminology is very important. You should be able to interpret the diagrams that are given in the question paper. You should be able to tabulate between the DNA and the RNA. You should be able to describe protein synthesis, and you should be able to do calculations. So there are calculations that normally are asked, and you should be able to do that. Then with meiosis, again, it is terminology, it is interpretation of diagrams. Again, you must be able to describe what crossing over is, when does it take place, and how does it take place. You should be able to tell the differences between meiosis 1 and meiosis 2. Also, you must be able to tell the differences between meiosis and mitosis. So this whole section in paper 2, it is the DNA code of life is 27 marks, and meiosis is 21 marks. So with the DNA and the RNA, we're looking at the structure, the location, and the functions of the DNA and RNA. Remember, you have studied all these things already. We are just reminding you, emphasizing the things that you need to focus on when you are studying, preparing for your end of the year. So the structure, the location, and the functions of the DNA and the RNA are important. That the fact that the DNA forms the genes which carry the hereditary information, you must know that. You must know what DNA replication is and why is it important. Why does DNA replicate, and why is it important? You must know DNA profiling, the uses, and how the interpretation of what of the DNA profiles. You must know what protein synthesis is, when does it take place, the two processes that are involved in protein synthesis, which is transcription and translation. So this is what you need to know when you prepare yourself, especially on this topic. So here we have the slide that shows the structure of the two nucleic acids. Remember, it's the nucleic acids number one, which is the DNA, and the second one, which is the RNA. So the reason we've put them side by side here, so that you can tell the difference between the two. When you look at DNA is a double-stranded molecule. You can see the two strands, whereas your RNA is a single-stranded molecule. And then another thing is on the DNA has a sugar. They both have sugar. They are both strands, and they are both made up of what? Of monomers, which we call the nucleotides. But then the difference is on the DNA, the sugar, which this part, this is the sugar. The sugar in the DNA is called the deoxyribose. Whereas in the RNA, the sugar, this one is called the ribose, hence the words RNA and what? And DNA. And then I said the DNA is a double-stranded structure, which is a double helix, a ladder. Just think of a ladder that is twisted. So that is why we talk about a double helix structure. And then the other thing is that your DNA, they are longer compared to your RNA molecules. So DNA are longer, RNA molecules are shorter, and then both of them are made up of the building blocks that we call the nucleotides. And then each nucleotide is made up of the phosphate. This is our phosphate. So both of them have this, which is a phosphate. And then nucleotide is also made up of the sugar. As you can see, both of them have the sugar. And then each nucleotide is also made up of a nitrogen base. So you can see here the nitrogen base. So these are the building blocks, and these are the three components that make up a nucleotide. Like I said earlier, we are not going to focus in the detailed content, but this is just a reminder of the things that you should know. And then with DNA comes DNA replication. We all know that we said DNA carries genetic information that must be passed on from this generation to the next generation, or from this offspring to the next. And then what happens is that that information, before it can be shared, it can be passed on, it needs to duplicate so that they are shared equally amongst the cells that are formed at the end. So with the DNA replication is when the DNA makes an exact copy of itself. So this one long twisted molecule will make a copy of itself, an exact copy of itself, and the process is called DNA replication. So what you see here, so when you study, you can take this diagram, you find it on the Mind the Gap. So this diagram, what you see here, it tells you the steps, but it does not give you the whole information. So when you look at number one, we are told that it unwinds. So it means that the DNA molecule does what? Because it's twisted, so it unwinds, it becomes straight. And then once it does that portion, what also happens is it unzips. If we go back, let me see if you can go back. If you go back to the structure of the DNA, you can see here, I said it is made up of two strands. And then the two strands are held together by these bonds here, which we call the weak hydrogen bonds. So they are held together by the weak hydrogen bonds. So when they replicate, those hydrogen bonds, they break. That is why then we say it does what here? It unzips. And then each side of the template of the DNA forms a what? It forms a template. So let's look at what I've just said now. We said the double helix structure, it unwinds. So this is number one. And then number two, the weak hydrogen bonds that are holding the nitrogen bases together, they break. And when they break, the two strands, they do what? They separate. So it unzips. The DNA unzips, and then it breaks and it forms two strands that unzip. And then once they unzip, it does what? They separate. And after separation, each original strand, this is the original strand. This is where we started. This side represents the original strand, and this side represents the original strand. So each original DNA strand serves as a template. You can see there, it serves as a template on which its complement is going to be built. So these are now exposed as templates. So what happens is after they are exposed like that, free nucleotides now in the nucleoplasm are going to, what they do is then they are going to attach on the original strand and the two new strands will be formed. So what happens here is, remember our nitrogenous bases, if you remember very well, we've got thymine, we've got adenine, we've got cytosine, and we've got what? Guanine. So if the strand is exposed like this, and this is how the nitrogenous bases are, then it means this strand is going to pick up nitrogenous bases which are going to complement this. When we say complement, so T, we always know that T matches with what? With adenine. So thymine always goes with adenine, or adenine always goes with thymine. And we know that cytosine matches with guanine, and guanine matches with what? With cytosine. So this is now the new strand that will be formed, and it complements the old strand. And when you look at the new strand that is formed, these two new strands will be exactly the same as the original strand. So then we said the two strands by attaching to their complementary nitrogenous bases, and we know that it's the A always attaches to T and C always attaches to G, or the other way around. And then this will result in two identical DNA molecules. So at the end, we're going to have two identical DNA molecules, and each molecule at the end, when you look at this here, the complementary bases are now formed. And then at the end, the two new strands that are formed here, they will look exactly the same as the what? As the original strand. So that is the DNA replication. So why DNA replication? Why are we doing this now? Because remember, there's another process that we talk about later in protein synthesis, which is more or less exactly the same as what? As DNA replication. And that question was in our 2022 prelim exam, where I expected to know the differences between the two. So let's look at the next process, protein synthesis. Protein synthesis, the name says itself. Synthesis, it's building something, making something. So this is the process by which proteins in our bodies are made. So that's protein synthesis. Protein synthesis takes place in two processes. Number one, it takes place in a cell. It starts in the nucleus, and then it finishes where? In the ribosome. So it takes place in two places, and it is in two processes. So process number one is what we called transcription, and process number two is what we called translation. So with the transcription results in the formation of what? Of mRNA. So in other words, the DNA provides a code. Remember those nitrogenous bases, the way they are arranged, T, A, G, C, that combination provides a code in the form of mRNA, and then mRNA is going to take that information to the ribosomes, where now the proteins are going to be built following this order that was provided by the DNA. So when we look at transcription, which takes place in the nucleus, sometimes you get a question like this and they don't give you this whole story, but you see a diagram like this. You should be able to tell this, okay, this is the nucleus, because you can see the nuclear membrane. This represents the nuclear membrane. And here we've got our nuclear pores. And the DNA, where DNA is found in the nucleus. So here it starts in the nucleus. What happens is, again, the double helix DNA unwinds. Remember, DNA replication, the same as this, but there are differences that you need to pick up. So again, here the DNA unwinds. And secondly, the weak hydrogen bonds between the nitrogenous bases, they break. And when they break, the two DNA strands unzip or separate. And here's the difference. Previously in the replication, we said the two strands, they act as a template. Here we are insisting, we're going to repeat this again. I know you are hearing it for the 10th time today, but we will repeat this until you go and write your exam. And then this one, in this case, only one strand acts as a template. Only one acts as a template for the construction of what? Of the mRNA in the nucleus. Then how does that happen? Instead of the DNA-free nucleotides attaching themselves on the DNA, the mRNA nucleotides will attach themselves onto the what? Onto the DNA. So we've got free DNA nucleotides from the nucleoplasm that will attach onto this one strand here. Remember, it takes place here. You said it unwinds and then it unzips and it breaks. Only one side, one strand acts as a template, not this other one. This part acts as a template, and then the free RNA nucleotides are going to come, float, and attach themselves here, and then the mRNA is what is going to be formed. So the mRNA complements the what? The DNA. So that side we said T, A, C, G, these are the nucleotides. And then with the RNA, remember the differences. The RNA, you need to remember that the complementary bases in the RNA, we do not have thymine. So where there is T in the, we are going to have what? Adenine on the RNA. But where there is A in the DNA, it is not going to be complemented by a thymine because there's no T, there's no thymine in the RNA, so it will be complemented by what? By uracil. And then the rest is going to be the same like in the DNA. So C complements G, or G complements what? C. So this will be the difference. So we are not going to say now the new strand here is exactly the same as the old strand, because it's not, because now there's introduction of what? Of uracil, which is only found in the RNA. So once the mRNA is formed, this is our mRNA, now it has been completed. It leaves the nucleus and it goes and it attaches itself on the ribosome. Here is our ribosome. This is the ribosome. And then when it attaches itself there, then these, there's things here. We call them the what? The tRNA. They are now going to bring in the anticodons, the base triplets. Okay, before the base triplets of the mRNA, we call them the codons, and the base triplets of the tRNA, we call them the what? The anticodons. So we talk about them as triplets. So it will bring these base triplets of the anticodons to match what has been brought by the what? The mRNA. And then doing that, they are bringing what? They are bringing amino acids. So when the tRNA comes here, it is carrying the amino acid with it. You can see the amino acids there. So these are now lined up according to the code that was provided by the mRNA. And then once they are lined up, this amino acid is brought, and then the next amino acid, the next amino acid, and then as they line up next to each other, they are joined to each other by a bond, which we call the peptide bond, and then a protein is formed. Remember, a protein needs a minimum of 20 amino acids. We can only call it a protein if it has 20 amino acids or more. So once they are lined up like that, and then they form a protein. So we said the coded mRNA leaves the nucleus and it goes to the ribosomes. Then the tRNA pick up the amino acids and complement the mRNA in the ribosome. Then amino acids bond by peptide bond and the protein is formed. So the difference that I was mentioning earlier is between the DNA replication and transcription. So note that those two processes are almost the same. The first two sentences are exactly the same. So what you see here in transcription is exactly the same as what you saw in replication. And then it starts to differ here when we say on transcription, one strand, on the other side, two strands act as templates. And then I said the other thing that is important is DNA profiling. Remember here, you will come across questions where you are given DNA samples. This is a DNA sample on the left here, on the right. Here, questions will be, we identify, they present a scenario where it's a crime scene, and then maybe fingerprints were left or blood samples were left there, and then the DNA samples of those blood stains that were found are then analyzed. And then in this example, we have the crime scene, and then there are 3 suspects. And then you need to identify now the suspects. You match with what was found on the crime scene, and you look at the suspect. So how does this work? It is a technique that is used to identify the sequence of bases. So we look at these bands, the dark bands here, the the nucleotides. These are nucleotides, they appear as dark bands. And then you're going to look at the original one, and then you look at what is presented as suspect or other 3 samples, and then you see how, if they match. For example, if you look at this, the second band here, we can't find it in suspect 2. We can only see it in suspect 1, it's not there, but we can see it number 2, but we don't see it in number 3. And again, when you do this, remember, you need your ruler. Right now, I'm just eyeballing it, and it's not easy. You need to have your ruler so that you can place it there and be able to identify where the bands appear across, or they appear more on this person compared to the rest, and then that is how you are able to identify. So here, with the DNA profiling, the nucleotides are separated from each other in the order that they are found in the DNA, or in the strand of the DNA, and then they appear as dark bands, and this information is used. We use it to identify in criminal cases, to identify deceased bodies that are not known. So it's got many uses. And the one thing that is important here, DNA profiling is not the same as, we no longer use DNA fingerprint. So avoid using that term when you answer this question. It is strictly called DNA profiling. It is not DNA fingerprints. So that is important here. And the uses of the DNA profiling are also very important. So what you need to remember in the nucleic acids, RNA and DNA, is the history of discovery of the DNA molecule by these people, Watson, Craig, Franklin, and Wilkinson. These people, they all played a role in the discovery of the DNA. And you must be able to tell the structural differences between the DNA and the RNA. Remember, we mentioned one has uracil, the other one does not. One is double-stranded, the other one is single-stranded, the other one is short, the other one is longer. You should also be able to tell the differences between the DNA, mRNA, and the tRNA. These are very important. And you should be able to differentiate between the DNA replication and protein synthesis, transcription and translation. You must learn these by heart. You've seen most of the question papers that you practice from. There's either 1 or 2 questions when you are asked to describe what transcription is or translation. And these questions are usually 5 or 6 marks. So that is why we are saying to you, learn these by heart. And then DNA profiling. Why is it important? Usually this one is going to come in the form of application questions. Like I said, you need your ruler. You need to read the scenario presented to you, analyze the samples that are given to you so that you are able to answer questions. Also, the paternity testing that involves both parents and the child. It involves both. When you talk about questions that talk about paternity testing, both parents should be involved and the child in how you respond to the question. And then the uses and the interpretation of the DNA profiling. So let's look at just one question based on the things that we've just done now. This was taken in the Free State paper that was written now in September. The question says the diagram below shows 2 molecules that are required during protein synthesis. So we are told it's 2 molecules required during photosynthesis. Remember, one tip that we always give you, when you get a question where there's a diagram, it is better you start by analyzing what the diagram is. What can you identify there before you go to the questions, so that by the time you get there, you already have some of the answers from the questions that are asked. So when you look at this diagram here, we can see that this looks like it's an mRNA, because this looks like it is a what? It's a tRNA, because here we see the anticodons, and then there at the top, I would say then this looks like it is a protein already. Protein is being built here because the amino acids are being joined together. So let's look at the first question. First question says identify molecule A. So what is molecule A? We said when we are just looking at the diagram, we said it is the mRNA. So let's see. The molecule A is called the mRNA, the messenger RNA. And then the second question is the bond at B. we said these are amino acids that are being joined to form a protein, and the bond that joins amino acids together is called a peptide bond. So let's see, it is the peptide bond. Number 3, the base triplet sequence for W. The base triplet, remember I told you about the base triplet, the codons and the anticodons, the ones on the tRNA, because this is the tRNA. So this should be the anticodons, okay? And then 1.2, name the organelle at which this phase of protein synthesis take place. So this phase is translation, because now the proteins, the amino acids are being brought based on the code provided by mRNA. And then we said this is the one that takes place in the ribosome. So the organelle in this case will be ribosome. Then name the nitrogen base represented by U. The nitrogen base represented by U, remember I said it's thymine, guanine, cytosine, uracil. Those are all the nitrogen bases that we've mentioned. So in this case, because this is the RNA, U is uracil. So that's C, that is uracil. So these were easy marks, easy 5 marks. You just study the diagram and you answer the question. So when they say study the diagram, you study the diagram. You underline what you think is important. You try and put in labels on the diagram, on the question paper, so that when you get to your questions, you've answered most of the questions that are asked. And then this one was taken from our Gauteng paper. Here we have a table that shows the DNA base triplets. So this is our table. These are the DNA base triplets that code for different amino acids. So that's table 1. Table 2 shows part of the sequence of anticodons that code for a polypeptide, which is a protein. Remember, I said a minimum of 20 or more, then we call it a protein. But if it's less than that, it is not yet a protein, but it is a polypeptide. Okay, so when we look into this, the question here says identify using the tables above amino acids 2 and 3. So here it means you are given the anticodons. So the one way of doing this is that you already have your DNA. So I always tell learners that you make a column of 3 a table of 3 columns. So you've got your DNA, you've got your mRNA, and then lastly you have your what? Your tRNA. So here you are asked to find the amino acids, which is number 2 and amino acid number 4, and you must identify them from where? From here, because we don't know their names. So amino acid number 2 is coded for by CCU. So we've got CCU, that is our tRNA, and then amino acid number 4 is coded for by UAA, that is our amino acid, our anticodons. So when we go back, if we work backwards, then the mRNA that matches with the CCU will be what? It will be A. And then the mRNA that matches the UUA will be U. So now we know what the mRNA looks like. And then we further work backwards towards the DNA. So here we've got GGA, then it means the DNA triplet that provided this code would have looked like C, and remember A matches with what? With T, so it's CCT. And then AAU here, remember again in the DNA A is replaced by T, so it's T, and then uracil. We don't have uracil in the DNA, but we have what? We have A. So now you've got the 2. So when you go back to your table, we are looking for CCT and we are looking for TTA. Here is TTA and there is what? CCT. So then it means the answer here should be glycine and alanine. So let's see the answer that is provided. Yes, it is glycine and alanine. So this is how you work it out. How many amino acids will be coded for by the anticodons in table 2? Again, you need to work it out like this so that you are able to go back and identify the. So here we are told that it is 3 or 4, because UUA and UUA, remember it's the same thing. So they will code for one protein, for one amino acid, and then these will code for different ones. So other learners might have said, it's 3 because this and this is the same. But some learners took 4 as its own and 1 as its own, and then we say that it's 4. And then name the describe the process that uses anticodons to make proteins, and that process is what is translation. So just like we mentioned it earlier, you need to describe how translation takes place, and as you can see here, it is 5 marks because the question said name and describe. So when you name and describe, you need to state that the name of the process is translation. That's where the first mark goes. And then the rest of the marks will go to how you then describe the process. So because of time, I'm just going to move fast to the other topics that we still need to do. And then with meiosis, this is the second topic. Meiosis, remember, it's the division where the cell, which is the haploid, undergoes a cell division, so that at the end 4 haploid cells are formed. So sometimes when we talk about it, we say it is a reduction division. The number of chromosomes is reduced, or the number of chromosomes is halved. So what is important here, you need to know what happens before meiosis begins. Before meiosis, we said what happens, DNA replication, which take place we call it a resting phase, which is called interphase, where the cell is not doing anything, but DNA replication is taking place. And then there's first division, and then the second division, and then there's biological importance of meiosis. So here, the terms again, important, you must know what your homologous chromosomes are, what bivalent means, chromosomes, chromatid, centromere, centrosome, crossing over, chiasma, karyotype, non-disjunction, karyokinesis, and cytokinesis. Mainly for which question on your question paper? Question 1.2, which is terminology. And with question 1.2, it is important that the spelling, remember sometimes when your teachers mark and when we mark, we say, no, we're not going to look at the spelling as long as it does not change the meaning of the word. But for 1.2, that is where we are strict. You cannot give the incorrect spelling. You are going to be marked wrong. So I said the resting phase between divisions is called interphase. I mean, that is where DNA replication takes place. So here, each chromosome consists of the 2 chromatids that are joined by what? By a centromere. What you see here is a chromosome made up of 2 chromatids and joined by a centromere. DNA replication takes place here so that the genetic material is doubled, so that it is shared equally during cell division. So here, there are terms that are going to come across your unreplicated and replicated chromosome. The unreplicated chromosome is this one. Before it goes into the cell division mode, when it's unreplicated, it's just one long string. Remember, we start from chromatin material, that mass of threads in the nucleoplasm, and then that mass of thread condenses and it becomes chromosomes, and this is the unreplicated one. And then further condensation takes place, and then we end up having a visible homologous chromosome or a replicated chromosome. So it looks like this. We start from that mass condensation, or it condenses and it becomes an unreplicated chromosome, and then it becomes a replicated. So the difference is that unreplicated is a single chromosome that appears in a cell before DNA replication takes place, and then replicated as it has double chromosomes with 2 chromatids. You see the chromatids there, and this is after DNA replication. So the phase with meiosis, important, you should be able to identify the phases. So whatever diagram that you come across, you must be able to tell that when I look into this is prophase. Why am I saying it is prophase? Because I see crossing over is taking place. I see the nuclear membrane is disintegrating. I see that the centriole is dividing and the spindle fibers are formed. So those are the things that you need to tell to write down. And remember, with meiosis, it is what you see. So you cannot say that it's prophase, but then you mention things that are not in the diagram. So here is a summary of everything, what you see in front of you. We have meiosis 1. This is stages of meiosis, but this is meiosis 1, and then the bottom part represents what? Meiosis 2. So the stages, as you can see here, we've got prophase 1, metaphase 1, anaphase 1, telophase 1. They are all characterized by special things that take place during each phase. So with prophase 1, what stands out with prophase 1 is that crossing over takes place. So you would see the chromosomes are lying next to each other. The homologous chromosomes lie next to each other, they touch, and it is only in this state where they are next to each other and they touch when we call them the what? The bivalents. So please remember that the bivalents, when these chromosomes from the maternal and the paternal side come together, lie like this, so that the crossing over takes place and they share. Remember, crossing over, what is important with prophase 1 is crossing over where genetic material is shared or exchanged between the two chromosomes, ensuring that variation takes place before that information is passed on to the offsprings. Because as you are an individual, you've received chromosomes from your maternal side and you've received chromosomes from your paternal side. But before those can be passed on to your offsprings, they should be mixed up. There should be that mixed up between the two, so that then they can pass on to, so that variation continues to take place. So we've got prophase 1 and then metaphase 1. How do you identify metaphase? The chromosomes will line along the equator. Avoid words like they lie at the center or they lie in the middle of the cell. We don't write it like that. It is not scientifically correct, but they lie along the equator. We call it the equator, the equator, the line. So that's metaphase 1. And how is metaphase 1 different from metaphase 2? At the equator, they are still in this form in metaphase 1. They are still in those pairs, the maternal and the paternal side. They are still lying like that. And then anaphase is characterized by what? We can see that they start separate, moving to the opposite poles. And then telophase, then the two cells are formed. The cells are formed. There's cell membrane constricts. It's called the invagination of the cell membrane. It constricts so that it separates, and then the two cells are formed. And then what you see here, we started with the centrioles moving to the opposite poles. The spindle fibers are formed, and then the centrioles are now at the poles. Spindle fibers are attached to the bivalents or the homologous chromosomes. Spindle fibers pull the chromosomes towards the poles, and then separation takes place. And then metaphase 2, the product of meiosis 1, meiosis 2, then continues with meiosis 2, product of meiosis 1. Then here, the two cells that were formed here, they continue with its division, so that at the end, how many cells are formed? 4 cells are formed, and each cell has half the number of chromosomes when you compare to the original. So here, important, you should be able to identify the diagram. You should be able to state why you are saying this is anaphase or this is prophase. Okay, let's just look at this one quick question before we move on. This was in our paper. I don't know if I must do this or the other one. Let me see. Okay, here we'll just skip because you've already seen this. You have revised with the teachers. The diagram below shows an error that occurred during meiosis in humans. We have one cell there starting with the 2 chromosomes. We see the structure 1 and structure 2. We see the membrane is disappearing. We see the centrioles here. So we can safely say that this is prophase 1 because the homologous chromosomes are lying next to each other. The centriole is separating. It's moving into opposite poles. So the question was, according to the diagram, which phase of meiosis did the error occur? So if this is correct, at the end, we were supposed to have 2 gametes, each having how many chromosomes? 2. But there's something different here. This one has 3 and this one only has 1. So it can only mean that there was non-separation somewhere. Because remember, after they lie next to each other to exchange genetic material, they should then separate. But so it means that they did not separate. They went together like that up until the last stage. Therefore, this and the separation takes place during anaphase. So then it means they did not separate. So that is why we say it happened in anaphase 1, because that's where they move to opposite poles. So which structure in cell A is the nuclear membrane? It's 2, because we see that it's disintegrating. And how many chromosomes are shown in the diagram? If you look at the first diagram there's only 2. State the origin of the chromosomes in cell A. Remember I said you get the chromosomes from where? From your maternal and from your paternal side. So that's exactly the answer for that. And then 1.4 said draw. Oh, that's another thing that I did not mention with meiosis. You identify, you state, and you should be able to state what happens in the next stage or what has happened in the previous stage. So here you draw the diagram of the phase in cell A before the error had occurred. We'll see that in the next slide. Then 1.5, in humans, how many chromosomes would be present in gamete 2? So if this is a cell representing a human being, remember a 46 chromosome, and gamete 2, because at the end of meiosis there's supposed to be 23, but because of this non-separation that took place, the expectation would be 22. So let's look at 1.4. 1.4 says draw a diagram of the phase in cell A before the error occurred. So the diagram of before this error occurred. So it means you are going to draw a stage where metaphase took place, because this is how you're supposed to draw. It's before. Remember, it's prophase and then metaphase. They line along the equator, and then anaphase where they separate. So we are drawing a diagram before the error occurred. So before anaphase, there's metaphase. So the question, because the examiners can't make things easier for you. They could have just said draw a stage showing a cell in metaphase, but that is easy. And so that is why then they say draw a one before, so that we can tell that you understand, you know what you're talking about. So the one before, then it's metaphase where they are lining along the equator. So you can see there. So what is important with our diagrams? Our diagrams must have a heading. You must have all your labels, and they must be clear. So here the marks were allocated in such a way that 1 mark went for the heading, and then the correctness of the diagram, that's another thing that we look into, and then the labels. So this is how you get marks, easy marks. If you know your captions, if you know your labels, and you make sure that your diagram is correct, that's how you get marks. So we are not going to look into this one, but it is found in a Free State paper. You already have that in your possession. So one of the trial exam papers that we've shared with you. I'm just going to go quickly through this. We are not going to do this because I want us to start the next topic before our time is not on our side. And then the last thing that you must remember with meiosis is non-disjunction. Remember that question that we've just done now of the separation and non-separation. That is non-disjunction, and what are the consequences of non-disjunction. So this happens when the spindle fibers fail to separate during meiosis. So when they fail to separate, remember it can happen in anaphase 1, it can happen in anaphase 2. The chromosomes are lying next to each other like this. So if they fail to separate, it means the two set is either going to move to one pole or the other pole, instead of separating and moving into opposite poles. So they fail to separate either during anaphase 1 or anaphase 2. And if it does happen on chromosome pair 21, now we are coming back to human beings because we always do application. So on chromosome pair in human beings, chromosome number 21, then it will lead to a formation of what? Of an abnormal gamete, because a normal gamete must have 23 chromosomes. So then that abnormal gamete will have an extra copy of the chromosome on 21. So 2 gametes receive extra copy of the affected, or 2 gametes will lack that chromosome. So this disorder in human beings is what results in what we call Down syndrome. So Down syndrome is caused by non-separation of the spindle fibers, such that the whole set of chromosomes move to one pole and then on chromosome 21 there will be an extra chromosome, and this results on what? On Down syndrome. This is what you just need to focus on. And then this diagram shows that it can happen here on meiosis 1. It can also, it can happen in anaphase 1. It can also happen in anaphase 2. With normal separation, this is what happens with normal separation. But with non-disjunction, as you can see here, both of them, they all went into one cell. So that is the end of the first topic. The second topic is genetics and inheritance. So with genetics and inheritance, again, it's terms after terms after terms. It's terminology. You must know your monohybrid crosses. You must know your principles, Mendel's principles of inheritance, your sex-linked inheritance, dihybrid crosses, blood groups, pedigree diagrams, and genetic engineering. When we say terminology, we're looking at things like, because sometimes we confuse chromatin and chromosome. Sometimes we confuse, so we must know what is the difference between the genes and the alleles. Remember, the alleles are just different forms of a gene. When do we say an allele is dominant? When do we say an allele is recessive? When do we talk about the phenotype? When do we talk about the genotype? What is homozygous, what is heterozygous, what is a monohybrid cross, and what is a dihybrid cross. So these are terms that you commonly confuse, and it's important that you know them by heart also. And then with Mendel's laws of inheritance, there's 3 of them. So this was taken exactly as it is from your exam guideline. So with the law of dominance, remember we say when 2 homozygous organisms with contrasting characteristics are crossed, then all individuals of the first generation will display the dominant trait. And then we also have another one which is called principle of segregation, where an organism possesses 2 factors which separate or segregate so that each gamete contains one of these factors. And then Mendel's laws, principle of independent assortment. This one says various factors controlling different characteristics are separate entities. So when they separate during metaphase or anaphase, or when they line along the equator, when they move during anaphase, all of that, they do not influence each other. It takes place independently. So they do not influence each other in any way, and they sort themselves out independently during gamete formation. So these are 3, and they are very important. We are going to take a 3-minute short break, and we are going to come back and continue with genetics. we're going to continue where we left off. We were talking about Mendel's laws of inheritance as very important. Remember, here you will get questions where you will be asked to state the law. Also, for you to be able to answer questions related to monohybrid crosses, dihybrid crosses, you should understand the Mendel's law of what? Of inheritance. So we said the law of dominance, when individuals are crossed with contrasting characteristics, we said that the individuals of the first generation will display the dominant allele. Remember, the dominant one is the one that always expresses itself in the phenotype. And then the law of segregation, where it says that an organism possesses two factors that separate or segregate so that each gamete contains only one of those factors, because remember, we've got those two factors from your maternal and your paternal side. And then the Mendel's principle of independent assortment, where I said that when they separate, when these factors that control this characteristic, when they separate, they do not influence each other. It occurs independently so that further variation is brought onto the cells. So then we're going to look at the format of a genetic cross. Again, I know you are sitting there thinking, we've had it like 6-7 times already. I'm repeating it again today. Remember, when a question says do a genetic cross to give a ratio or to give a proportion or to tell us what will the phenotypes look like, this is how the layout of a genetic diagram looks like. We are telling you this because by just putting down these steps before you even put the actual letters or the physical appearances of whatever you are discussing or you're talking about, you get how many marks? You get marks. There's marks for the P1 and the F1 generation. There's marks for meiosis and fertilization. We call it a formula. Some teachers, they call it a template. We give it so many names, but it is very important that you know this. So the phenotype, those are the visible trait. So when you are given a genetic question where you're supposed to do a cross, you first look at the phenotype. What is it that they are telling you? Are they telling you that it's black or white or it's green or red? That is the physical appearance. That is what you put as the phenotype. And then you go on to look at the genetic makeup. So sometimes you will be given the letters to say, okay, brown is represented by letter B and black is represented by small letter b. So you've got your capital letters and small letters. That is your genetic makeup, which is what we call the genotype. Remember, the genotype influences the phenotype. It is not the other way around. So it's the genetic makeup that tells us how the person is going to look like, or how the plant, or how the flower, the color of the flower is going to be. And then the alleles segregate during meiosis. So remember, law of segregation there. During meiosis, the alleles separate. So that is what you show here, separation, and then you show the gametes. And then fertilization takes place. Then this results into what? Into your phenotype, which is F1, and your genotype. So what I've shown that side can also be represented in a form of a Punnett square. So you can do the cross like it appeared in the previous slide, or you can do a Punnett square. Most of the learners prefer a Punnett square because it's easy and I think it's quicker to do than the other one. So this is your format of a genetic cross. It is very important that you indicate, and your teachers always say to you, everything must be in its line. It makes it easy for you to see what you are writing. It also makes it easy for the person who's going to be marking you to see that you've put everything in its correct order, and you get your 6 full marks. Remember, this whole thing is just 6 marks. So remember that by writing P1 and F1 and meiosis and fertilization, you already have how many marks? 2 marks in the bag. Then the genetic, the steps that you should look into, or things that you should take note of when you solve or when you're going to do a genetic cross, is number 1, you must identify the phenotypes of the parents. I said earlier, you look at things like the color, if it's mentioned, or the height, if it's mentioned, anything that refers to physical appearance, that's the phenotype. Secondly, you choose the letters to represent the alleles. Sometimes letters will be given, sometimes they won't be given. You'll have to come up with your own letters. And then you look at you identify the genotypes of the parents. So if they are talking about homozygous black or dominant this and recessive this, if there's dominant this, recessive this, then you know that the dominant we represent it with a capital letter, and the recessive is always represented by a small letter. So if it's homozygous dominant, then it means both alleles are the same there, then you have your TT. If it's heterozygous tall, and then we know that it has got one dominant allele and a small recessive one. That is why we say it heterozygous. So homozygous can be homozygous dominant, can be homozygous recessive, because these two for recessive can also be paired together. And then you show how the alleles separate during meiosis into gametes. That's what we've shown in the diagram there. And then you show all the possible genotypes for the first filial generation and when fertilization occurs, and then you also interpret the phenotypes. So you start with the genotypes, it will then help you interpret the all possible phenotypes. So when we talk of monohybrid crosses, remember monohybrid crosses are the ones that involve one cross, whereas dihybrid crosses, not one cross, but one trait or one characteristic. Whereas your dihybrid cross, we're looking at the two traits or the two characteristics. So we have complete dominance, or the types of dominance. We have complete dominance, where only one parent's phenotype is evident in the offsprings. And then we have incomplete dominance, where neither of the parents' phenotype is evident in the offsprings. So for example, here we're looking at the homozygous green crossed with the homozygous yellow. But in this case, green is dominant over white over yellow. So all the offsprings that are formed, their physical appearance is what? Is green. So this shows what? Incomplete dominance, that the green was dominant over white over yellow, so they are all green. With incomplete dominance, neither of the parents is evident. In this case, a white flower is crossed with a red flower, and all the flowers are what? Are pink. So that is incomplete dominance. And then with co-dominance, it's when the traits, we say they coexist. They show themselves in the phenotype. So in this case, a white cow crossed with a red cow, we have a color that is a mixture of red and white. And this new color is now called what? Roan. We also see this in the blood groups. Remember, when you get there, your blood type A and B, those are also dominant on their own. So when they come together, that is why we've got people who've got blood type A, B, because it can neither be A or B, because they are both what? They are both dominant. So these are the three types of dominances: incomplete, complete, and co-dominance. Know the difference, because you will get questions where you need to be able to tell between the three. And then with the genetics, again, there's also what we call sex determination. Before we do that, remember, when we look at the cell and how the chromosomes are displayed in the cell, or how they are laid out, we talk about the karyotype, which represents the chromosomes in a cell. And in humans, the female karyotype is different from the male karyotype, in that if you look at them, okay, first of all, we have 23 pairs of chromosomes, or 46. So these are all the pairs up until chromosome number what? This last one is number 23. This is in the male. This last one in the female, also 23. If you look into the two, in the male, there's one big chromosome and one small one on the last pair, whereas in the female, they are both equal. So in the males, we have what is called the X and the Y chromosome. In the female, we have both X, two X chromosomes. So the sex chromosomes, we call them the gonosomes, and then the other ones, all the other 22, they are what is called the autosomes. So you have the autosomes number 1 up to number 22, and then we have this last pair, which is called the gonosome. So your autosomes are 1 to 23, and your last one, chromosome number 23, is called the gonosome. So if this is presented in front of you, you should be able to tell the difference with this is the male and this is the female. And the reason is you will say that chromosome pair number 23 for this one is both chromosomes are the same, therefore it is a female. Or chromosome pair number 23, one chromosome is longer, the other one is shorter, therefore it means it is a male. And then with sex determination, what we say here is that there's a 50% chance of having a child that is a boy or a girl, because remember, the females, they are carrying two X's and the males are carrying the X and the Y. So in whatever happens during fertilization, when the gametes are produced, the females are always going to provide an ovum that has an X, whereas male are going to produce sperm cells which have either X or Y. And then when the combination between the two takes place, that is when we say 50%, because this does not mean that if your parents happen to have 4 kids, then the expectation is 2 of them are going to be girls and 2 of them are going to be boys. No, it means that each time fertilization takes place, there are 50% chances that that child is going to be a girl or a boy. So this is how then sex determination is explained. And then we also have what is called the sex-linked disorders. Remember, your chromosomes, they carry genes. Genes are found in all your chromosomes. So there are also genes that are found on the sex chromosomes. So in whatever disorders caused by the genes that are found on the sex chromosomes, those are the ones that we call the sex-linked disorders. So fathers can pass the X-linked allele to their daughters, but not sons, because remember, the X determines the female and the Y determines the male. So here they say fathers can only pass the X to the daughter, because the boy will get its X from where? From the mother, because the mother passes their X to both the boy and the girl. And then one of the disorders or sex-linked disorders that are in your exam guideline that you must know is the inheritance of hemophilia, and the other one, which is color blindness. Because these two are in your exam guideline, these are the two that you are supposed to know. Sometimes you'll find that in an exam, you are not going to be given all the information about hemophilia, because the expectation is that you know it, you learned it in class. But if it's a new sex-linked disorder that you've never seen before, the examiners will make sure that they will give you all the information so that you are able to answer the questions. So here again, we say the father can only pass the affected allele to its daughters, because remember, father can only pass the X to the daughters, but the mother can pass the affected allele to both the sons and the daughters. So this one is caused by the recessive allele, which is normally represented by what? By the small letter H, and it is found where? On the X chromosome. And then the reason why we focus on the X, because the Y chromosome, as you saw on the karyotype, they are small. So because they are very small, they don't normally carry genes. There are no genes that are found in there. So all these disorders that we talk about, they are mainly found on the what? On the X chromosome. So here, this cross shows a father without hemophilia and the mother who is a carrier. So the fact that the mother is a carrier, it means that mother has capital letter H and what? And small letter H. And then the father only has a capital letter H and Y, because the father does not have hemophilia. So the result then will be the son with hemophilia, because the son might have received this small h from the mother, the daughter who is the carrier, because got again the small h from the mother, and the son without hemophilia, because happened to receive this h which is not affected from the mother, and the daughter who does not carry a hemophilic gene, because the daughter received the h from the father, the big H from the father, and the big H from where? From the mother. So that is how you work it out. Same thing with color blindness. It is also a sex-linked disorder that where it affects the eyes, such that you, especially male, they are not able to tell the difference between the colors. Again, here it's the same explanation as the one that I did earlier. So please know your hemophilia and know the color blindness. And then the blood groups, here the inheritance of blood groups, remember we said all the characteristics are controlled by 2 traits. But for genes, they are controlled by what? We talk about the multiple alleles because there's about 3 of them. And then here, what you see on the left-hand side are the blood groups, the phenotypes, which is what is seen by the doctors when they check what blood type are you, and then the genotype. So we've got blood type A, B, and O. These are the blood types that are available out there. And then the genotypes. So when you write the genotype, you are not going to write A, and say this is the genotype. We are going to mark you wrong. When you write the genotype, remember, it's the I and the superscript A, capital letter at the top, I and the superscript A. This is the genotype of A, because then this person is homozygous for A, or this person has only one dominant A and the other one is the recessive I or O. And then again here, because A is dominant, A is going to show itself. So that is why we talk about only blood type A. Same thing applies to B. Because B is dominant, B is always going to show itself. But where there is A and B, both of them are going to show themselves because they are what? They are co-dominant. And then O is when it's the recessive allele. So O is a recessive allele. It will also show itself if you've received O from your father and O from the mother. Then you will have blood type O. So I said here, that is where we see for A, because I said A is always dominant and B is dominant, so they show incomplete dominance when you talk about the type of dominance. But A and B shows co-dominance, and then O also shows what? Complete dominance. Then another thing that you must also remember here are your genetic lineages or pedigrees. These are very important. I know that we've said before, in future, you are not going to be provided with us the keys. Then you must know that if the circle represents the female, the square represents the male. If it's shaded, it means it is affected. If it's not shaded, it means it is what? It is not affected. And then you must also know that these, like you can number them, you can say this is 1, this is 2, this is 3. And these do what? They represent generation. So we look at the first generation, the second generation, and the third generation. So there's 3 generations represented by this pedigree diagram. So it traces the inheritance of characteristics over many generations. So if you need to find out how did I end up having a son or a girl, that a daughter that has hemophilia, whereas mhlambe it did not show in both parents, then when it's traced back using this, then you can pick it up that it was in the grandparents. That is where the gene came from. So with understanding of the pedigree diagrams, like I said, female is a circle, male a square, affected is shaded, and then the line shows the marriage line, which is when it's like this, and then if it goes down like this, it's the line of what? Of descent. And then if it's represented like this, it represents the what? The sibling line. So it means these two are siblings, whereas these two are married, and then they gave birth to the two there at the bottom. And then the steps that you must know involved in pedigree diagrams. The key, it's in your Mind the Gap, it is in your SIB material, it is also in your DBE book, the green one. So if you go through all the steps there, you will know. You always look for the dominant and recessive alleles. You always look for your letters, and then you try and fill in the letters throughout, so that when you get to the questions, then you can answer. You look for the dominant and recessive characteristics, you fill in the genotypes, and you look for the ones that have a recessive conditions. Where did they get the recessive allele? And then you work backwards and you fill in your recessive alleles for each parent. And then if parent has a dominant characteristic, you put that one in a capital letter, and then that's how you work out a pedigree diagram. Again, you see what paternity testing. We talk about paternity testing in the DNA profiling. Again, it comes here that 2 things can be done for paternity testing. It's the blood groups and it is the DNA profile. But the most reliable one here is the DNA profile. It is not this, because with the blood groups, we say there are many people who have blood group A. There are many people with blood group B. So we cannot really say that this is the father of the child because they've got the same blood group. Many people share the same blood group. But with the DNA profile, that is where you can really tell who is the father. So this was one question that was in our question paper, which talked about the cheetahs that are carnivorous found in the dry grasslands and woodlands of South Africa. And they normally have the spotted fur, but however, a king cheetah has a rare variety. We've got this one that has inherited the striped fur. So we're looking at the two things here, the striped, we're looking at the spotted. So while declining cheetah population, with the declining population, a breeding program was started to increase the number of cheetahs to introduce to the wild. So using the breeding pairs, 8 cubs were born. The first breeding pair produced 4 cubs, and the second breeding pair produced 4 cubs also. And then the results are shown. So we've got 2 traits. We are already seeing 2 physical appearances, the striped one and the spotted ones. So this is the table. So the question here says, using the alleles capital letter N and small letter n, state the possible genotypes of the striped cheetah. So the genotype here is small letter n and small letter n, because we're looking at the table that is given here. So here a spotted is crossed with with a spotted, and the result is that 3 are spotted and 1 is striped. And then the second one, a spotted and the striped. So here it tells us that 2 with spotted fur and 2 with striped fur. I'm going to stop here so that I give my colleague a chance to do evolution. And another thing is that yes, so I'm going to stop here and we'll start with evolution.
Welcome back, grade 12s, to this lesson revision of paper 2 for life sciences. We're going to get right into evolution. First, I want to apologize for that break. We had some technical difficulties, but we are right on board. So let's start with evidence for evolution. But first, I want to remind you that evolution is a very large portion of paper 2 and a very important one as well. And also the place where you can score a lot of marks. I'm going to give you those tips for you to show you where you can score those marks and just highlight some very important information. Firstly, something that seems small but not small at all, the evidence for evolution. You need to know that the evidence for evolution are biogeography, which basically means that if we have organisms that look similar and have similar characteristics, but they're on different continents, that at some point that must have meant that they shared a common ancestor. And usually biogeography is related to continental drift, where we had a continent and then they separated during continental drift. We had geographical barriers between them, which are the oceans, and then we had formation of new species. We'll talk about it a little bit later. Then the second one is genetics. This is the genetic makeup of an organism and how mutations occur, random mutations, which actually contribute towards evidence for evolution. Homologous structures is when we have different organisms that have different functions for their limbs, but the anatomy of their limbs are similar. So they have similar anatomy, but they have different, but that specific limb have different functions. That's also evidence for evolution. Lastly, the fossil evidence. This is solid evidence. The fact that we have fossils that are found from millions of years ago and they look different from species that we have today. That gives us also evidence that organisms have evolved over time. Right, so what are the sources of variation? So variation is how we differ, right? So you should know the sources of variation. Nonhlanhla went through it nicely with meiosis, the first two, crossing over and random arrangement. Just to remind you, crossing over happens during prophase 1 of meiosis and random arrangement during metaphase 1 and 2. They have random arrangement of chromosomes, right? These two contribute to variation. Then for reproduction, we have random fertilization and random mating, which contribute to variation. And lastly, we have mutations, these changes in the DNA sequence, which can contribute to variation of an organism. I've just got a mnemonic there for you to remember, CRRM, to help you remember these sources of variation. As soon as they say you can start writing in your question paper, you quickly write down all of these mnemonics and all these little things that you try to that you remembered when you were studying, these little relationships you made to remember your work. Then we come to types of variation. This is different from sources. Sources is how the variation came about. But the types of variation, we get two. We get continuous variation and discontinuous. This picture, this first picture shows you excellently the height, skin color, and hair color, how they differ and how there's a range of them. It's not just dark and light skinned. It's not just dark and light hair or red and blonde hair. It's not just tall and short. There's a range of different phenotypes. Whereas discontinuous variation, this is when we have specific categories of phenotypes. For example, you are either blood type A, blood type B, AB, or O. Right, now we move into the theories of evolution that you need to know. The first one is Lamarck, and he's first because it was in the early 1800s that he basically discovered these two laws. The law of use and disuse and the inheritance of acquired characteristics. You need to know both of them. You need to know the two laws, and furthermore, you need to understand how to apply the law to a specific question. So just to guide you to the first 2 points I have over here, the difference between an inherited characteristic and an acquired characteristic. An inherited characteristic is something that an offspring is born with. Okay, it's a characteristic that we are born from and we get from our parents, and it's controlled by a gene. Whereas an acquired characteristic is something that the offspring is not born with but develops in their lifetime. and it is not controlled by a gene. For example, if you are working out, okay, and you are building muscle, then you are going to be lifting weights and you're going to be building muscle, and that muscle that you've built up is a characteristic that you've acquired, but it's not a gene. It's a characteristic that you required and you acquired, and you cannot pass it down to offspring. Okay, then these guiding points over here can indicate to you the questions. When Lamarck's theory is applied to a new situation, these are the questions that you need to ask yourself, okay, to help you through this type of questioning. Just to go back quickly on that, to just use the thing about the giraffes, just to remind you, what did he basically say? He basically said that all giraffes had short necks, but because they wanted to stretch and they needed to stretch their necks to get the trees, the leaves from higher up on the trees, they had to stretch their necks. And because they continue to stretch their necks got longer and longer. And then they passed down that acquired characteristic to their offspring. And that's how all giraffes have tall necks today. But something sounds a little bit off about that, right? And you've learned that that was now rejected, that these theories were rejected. And the reasons why is what you need to know. You need to know that there was no evidence that acquired characteristics are inherited by offspring. Organisms did not evolve because they were determined to change, but these changes actually happened due to random mutations. That's a very important concept for evolution, that these things happen due to random mutations. An organism cannot change its DNA, and according to Mendel, it is the genotype that determines the phenotype and not the other way around. Right, then we come in 1859, when Charles Darwin came about and published a book, and he had a theory of natural selection that had two main points, and you need to know these. Number one, species were not created in their present form. So the species that are in existence now, they were not created in that form, but they evolved from ancestral species. So in other words, there was a common ancestor, and over millions of years, species adapted and changed. And second, he proposed the mechanism for evolution, which was called natural selection. Right, natural selection is one that you need to know. It is in the examination guideline, bullet for bullet. Learn it off by heart. That is your recipe or your template for natural selection. If the question has to ask you, describe Darwin's law of natural selection, you're going to write those bullet points from your examination guideline as is. However, if they give you a scenario, in this particular one, it's about lizards on an island. If they give you a particular example, you need to read the information in there and then look at this question. It's saying, use Darwin's theory of natural selection to explain the evolution of lizards with larger jaws. So they're specifically asking you to talk about the lizards and their jaws, okay, and how they eventually had larger jaws. So you need to apply it to your natural selection recipe. But let's quickly just go through the recipe quickly. The key words that you need to remember when you are doing natural selection is that your first bullet point has got to do with variation. There was variation in the population. Right. Then you mention the favorable and the unfavorable characteristic. You do not say there were favorable and unfavorable characteristics. You need to apply it to this question. Okay, but that is the next keywords. There were favorable and unfavorable conditions. Then favorable and unfavorable characteristics. Then you mention what happens to the unfavorable characteristic. The organisms that have the unfavorable, then they die out because they cannot survive. They don't have the favorable characteristic. And the ones with the favorable characteristic, they survive and they reproduce and they pass the allele for that characteristic to their offspring. Okay, that's generally the recipe. So in this particular one, we needed to apply it to the specific lizards. Right, so we're starting with the first bullet point. Remember I said variation. So firstly, there is variation in the size of the lizard jaws, not just the lizards in the jaws. Some had small jaws and others had large jaws. These are the favorable and unfavorable characteristics. This is the favorable characteristic and the unfavorable characteristics that were mentioned in the passage. Due to the larger supply of fibrous plants, those with smaller jaws were unable to feed because they couldn't eat those fibrous plants. Therefore, they died. But the lizards with the larger jaws, they were able to have more food and they survived and they reproduced, and the allele for larger jaws was then passed down to the offspring. The next generation will then have a higher proportion of lizards with larger jaws. Okay, so do you see how it is different from what Lamarck said? Right. So that is basically natural selection, where nature was a selective force. The food that was available there was the ones who survived were the ones who could eat the food that was available. Right, then we have another theory called punctuated equilibrium. In this theory, it basically is saying that we had an organism and then we have periods of a rapid change. Okay, so you can see this one went from being beige to being white. So we had periods of rapid change and then we had periods of equilibrium. So you can see that this whitish colored mouse over millions of years or over a long period of time still remain that color. So we have some long periods of no change or very little change, and we have rapid periods of a change. Here we also have a rapid period of change where it just went from this beige color into this brown color. Whereas gradualism's idea is that species occur as a result of a series of small changes. It goes from a beige mouse to a little bit lighter to lighter, and then we have another branch over there that becomes the darker colored mouse. But you can see that the colors change gradually. Right, another important one that I'm not going to spend too much time on is artificial selection. The main thing that you need to understand about this is that it's the intentional reproduction of individuals in a population that have desirable traits. Okay, so this is when humans are going to breed specific organisms because they want a desired trait. For example, in domesticated animals, mostly it's because for food security, we want bigger chickens, we want more chickens, right? And then an example for a crop species would be this one over here, where we've got a mustard plant, and then it was selected for specific characteristics so that we can get what we know today as cabbage, Brussels sprouts, kale, broccoli, cauliflower, etc. Right, so you need to know those. You need to understand that what is the purpose of this, and the purpose is that humans are the ones that are the selective force, not nature, and humans are the ones that choose the characteristics which are desirable for what they need. Right, now before we move on to speciation, which is another type of theory that we need to know, I need you to first understand these terminology because learners commonly confuse them in the exam. And if we don't understand these terms, it's difficult for us to understand speciation, and then we lose many unnecessary marks. Right, so let's first start with the species. I've got a picture over here to assist us. A species, they are organisms that have the same characteristics, capable of random interbreeding to produce fertile offspring. So here we have an example of this organism. Okay, so it's organisms that look like this one. They have the same characteristics. They're capable of breeding and interbreeding and produce fertile offspring. A population are individuals of this same species. So it's this organism over here, a group of them living in the same area, and they can also interbreed. Okay, so this picture over here is showing us a population. Can you see that they are the same organism? They are the same species. Now, what is speciation? Speciation is an evolutionary process by which populations evolve to become distinct species. Okay, so this is an evolutionary process where they actually become species. And how does this happen? It happens by either geographical isolation and reproductive isolation mechanisms. Basically, these two, they isolate the gene pool of a species, and it results in the formation of a new species. So let's look at an example of this. Okay, we're first going to look at speciation, and then we're going to look at reproductive isolation. Right, so the first one is speciation. Once again, there is a recipe in your examination guideline. Learn it off by heart. Okay, but almost always they are going to give you an example. When it comes to speciation, they're going to give you a specific example that you need to apply your speciation description. You need to apply it to the question that is being asked. Okay. So let's start with this one. We're using, this is the generic answer, the generic answer that you will actually find from your examination guideline. So I'm just going to go to the next one. That's actually a specific example that's also going to explain this. In the question, they're telling us that the present day distribution of 3 closely related species of the dog family, the coyote, jackal, and dingo, is shown on the world map below. Okay, so here they're showing us. Now, this is already, does it ring a bell to something I said earlier about biogeography? Doesn't it look a bit similar, this question? Yes, because they are showing us that the coyote, jackal, and dingo are living on different continents, right? But they tell us that's the present day distribution. But they're telling us that they're closely related species. So this has to give us an indication that, okay, biogeography must be the evidence here, because this is basically telling us that there must have been a common ancestor that was once living on a continent and then it got split. So let's see how we look at that question. Firstly, what is the type of evidence? It is biogeography. How do we know that? It's because they showed us the different continents, as I said, and they showed us the species are on these different continents. But they share similar characteristics, and they share similar characteristics. Describe how these 3 species could have evolved from a common ancestor. So do you see they're not even mentioning the word speciation? But you must know, firstly, the mark allocation can also indicate to you that it's going to be a long description that you're going to need to give, and that they want to know how they could have evolved, the species evolved from a common ancestor. That's going to give you the indication that it is speciation. So let's have a look at the answer. You first need to mention that there was a common ancestor that lived on a large continent, okay? And then they became separated by continental drift. In this particular case, it was the ocean. And there was no gene flow amongst these 3 populations. Now, this is where I want to stop. It's very important that we say that there was no gene flow between the populations. And it's also important that we say that the population got separated, or the population got split, because it is not the species, right? We are humans, for example, are all part of the same species. We are all Homo sapiens, right? But we live in a population. So if something has to happen to us, and we are split by a geographical barrier, it's not our species that is split. It is the population of all of us that gets split into another 2 populations. And in this instance, they got split into 3 populations. Each population experienced different environmental conditions. What I see learners say is that they just say there were different environments. The individuals went through natural selection. Okay. But it's important that we understand that it was each population that experienced the different environmental conditions, and they underwent natural selection independently. The individuals in each population became different. Right, it's important that we say that, because on either side, in each population, they had different environmental conditions. So they went through natural selection independently. So they individually became different, right? Phenotypically and genotypically. Even if the 3 populations were to mix again, they would not be able to interbreed, right? Or to produce fertile offspring, forming the different species, the coyote, the jackal, and the dingo. Please take note of the compulsory marks. You're not going to be able to know where the compulsory marks are, okay, before the time. The only advice I can give you there is that you need to be as specific as possible to the question that is being asked. Apply your knowledge to the question that is being asked, and make sure that you're answering the question, okay? Because these marks, these compulsory marks, are directly related to what the examiner wants you to answer from the question that he or she has given you. Right. So the main thing over here is to know the recipe, Keep your ducks in a row, know when to use species, know when to use population, understand what happens between them, and be specific to the question that's given to you. Right, now we've got mechanisms of reproductive isolation. This also contributes, but these ones, the specific ones you need to know are breeding at different times of the year. For example, we can have these butterflies over here. They breed at different times of the year. So they are different species and they breed at different times of the year. So this prevents them from actually interbreeding because they are not ready to breed when the other one is. Then we have species specific courtship behavior when we have, like mating dances and we have courtship dances and which organisms do to attract mates. So some mates are not mates of other species will not be, organisms of other species will not be attracted to that particular courtship behavior, right? So, breeding will not happen, so that also prevents it. Plant adaptation to different pollinators. This, for example, is because they are physically different shapes. So we have different shaped flowers, for example. So only certain birds or insects are able to pollinate them because it depends how they fit. Then we have infertile offspring where we have two organisms that are different species. They do manage to interbreed and they do manage to produce offspring, but that offspring is not fertile. That's this picture over here. And then we have the prevention of fertilization when the reproductive organs do not fit or they are not compatible. Right, evolution in present times. Basically, this is rapidly reproducing species which allows us to study. Okay, no, let me start again. Evolution in present time. This is how natural selection, example of how natural selection is actually happening around us. Okay, an example of this is the Anopheles mosquito with malaria. We have the COVID-19 virus, which we are still overcoming. And what you can see from it is that it's mutating all the time, okay? And it's changing. It's changing, it's getting stronger, it's mutating, and HIV. So just a quick example for of how to explain this to you. You basically you apply natural selection to this. So for example, you have a population of mainly susceptible bacteria, and when you apply the antibiotics amongst this population, there are still a few that are resistant, okay, to the bacteria, and those will survive, and the ones that are not resistant, they will die. And eventually the ones that are resistant to the bacteria will survive and reproduce. And when they survive and reproduce, they pass that allele down, and then you eventually have a population of resistant bacteria, because the ones that were not resistant, they had the unfavorable characteristic, they died when the antibiotic was administered, and they basically died out. So does this ring a bell? It should ring a bell because we spoke about it earlier when we did natural selection. So this is just another place where you can be asked to talk about natural selection. You follow the same way. They also like to ask this question in scientific investigations. Right, now moving on to human evolution. These are the possible questions. I'm going to pause here a bit. This slide is very, very important. These are the possible questions that you can be asked in human evolution. So we've done the theories, we've done the sources of variation, and we've done speciation and natural selection in particular. Now moving on. We have interpretation of phylogenetic trees. We have the similarities between African apes and humans, so what we share with them. The differences between African apes and humans, specifically looking at the skull, bipedalism, and that bipedalism, what contributes to that is the position of the foramen magnum, the shape of the spinal cord, and the pelvis. Then we have fossil genetic and cultural evidence. You need to know that, and you need to know the fossils that were found in South Africa and the scientists who found them. And then lastly, the out of Africa hypothesis, that you need to know exactly what it says in the examination guideline. Right, so let's start with the phylogenetic trees. These are the questions, this speech bubble here is very important. These are the questions that you need to ask yourself. right? Or the things you need to know when you're looking at a phylogenetic tree. Who is the common ancestor? Who is extinct? When did a species on the diagram originate? How long did they live for? And when did they become extinct? Now, grade 12, phylogenetic trees is a very important question. Examiners like it. Okay, they like asking this question. It is a skill, as you've mentioned many times before. And you just need to remember those few things. So the few things that we need to look at and that we need to remember is that when you are using a reading from a phylogenetic tree, you must use a ruler. You must identify where is the past and when is the present, and I've already indicated it here. This is the past because it's so many million years ago, the bigger number, and then 0 would be the present day. Okay, then what was the other question they asked? Who is the common ancestor? So where will you find the common ancestor? It's the oldest one that you can basically find, the oldest one where we have the first branching. So you look for the original, how far back it goes, and the first branching off, and then here it will be at this point, which is the common ancestor. The ones branching off from the common ancestor are the descendants. Okay, and then they want to know, okay, who is extinct? So if you have a graph and in the middle of the graph all of a sudden the line stops, like over here for example, that means at this point that organism, that species which was A. robustus, became extinct. Now the question will ask you, how many years ago did A. robustus become extinct? You take your ruler, you draw a straight line. I don't have a ruler here, so mine's not going to be so straight. You draw a straight line down until you reach the x-axis, and you read the value off there. Now, where learners go wrong is that they don't remember or they don't realize that the graph is going 0, 1, 2, 3, 4. So this number is between 1 and 2. It is not 2.5. It is around 1.4, okay, or 1.5. It's very important that we know how to read this, okay? Another thing is that they can ask you, when did it appear, or when did it arise? When did the organism appear? And then it would be, for example, at this point here, okay, you go to the start of that line, and once again you draw the line down. Then they can ask you, how long was A. robustus in existence for? Then what do? You take the big number, you measure this whole line between this point and this point. When did they appear? Minus when did they become extinct? And the answer will be in million years. That's how long that they were there for. Now, you won't get phylogenetic trees always in this way, in this graph. Sometimes you read them from left to right. Sometimes you read them from the bottom up. Sometimes they give you a line. Sometimes they give you a block to indicate the species. You just need to sit down and have a look at what they are giving you and what the question is that they are asking you. Right, very important question, the similarities between Homo sapiens and primates. And we have a little man or lady there for you. Actually, it's someone that has both reproductive organs, right? And then here we have, I've just color coded it to help you understand better. And when you're doing this, you can even look at think of your own bodies while you're in the exam to ask yourself, okay, what are the similarities here? So let's firstly look at the head region, right? We have large brains compared to their body mass, okay? We share this with the African apes. We have eyes in front, right? We have binocular vision, stereoscopic vision. Right, now let's look at the arm. We have freely rotating arms. Okay, you can physically do this in the exam to see that, this, to remind yourself. We have longer upper arms. This is the upper arm, right? We have longer upper arms. We have elbow joints allowing rotation of the forearm. So our forearm is able to rotate. We have 5 fingers and we have an opposable thumb, which works in opposite direction to our fingers. And then we have flat nails instead of claws, and we have bare fingertips with fingerprints. We have 2 mammary glands, and then we have, that's part of the face, we have eyes with cones or color vision. And lastly, we have sexual dimorphism. Okay, we have males and females separate with their own sexual organs, reproductive organs. Okay, it's color coded. You need to know this. You have your own diagram. Use your body the way I did to help you remember these different things. Right, now we have the differences, also important. They like to ask you to tabulate. When they ask you to tabulate grade 12s, remember that you must draw a table. You just draw a table like that with your ruler, nice box with the columns and this division, and you get 1 mark for the table. Okay, and then when you're comparing, you must compare like things. If you talk about the cranium on this side, you must also talk about the cranium. So let's first look at bipedalism and how it contributes to the differences in humans. The bipedalism in humans and quadrupedalism in African apes. What does this mean? Commonly also where learners make a mistake is that bipedalism, it's important to say that they walk on 2, it's the lower limbs, right? You don't just say legs, or because it could mean anything. So you must refer to the lower limbs. And quadrupedalism is when they basically are able to move on all 4. So what contributes to this? And it's something that you need to know. The foramen magnum is in the forward position in humans. You need to use those words. And the foramen magnum is towards the back of the skull, or in a backward position in African apes. The shape of the spine, we have an S-shaped spine in humans and a C-shaped spine in African apes. With bipedalism, humans have a shorter and wider pelvic girdle. Grade 12, these are the words you use, and only these words. In the African apes, longer and narrower pelvic girdle. Learn these words. It's the only ones we will accept. Cranial size, it's a larger cranial size in humans and a smaller one in African apes. The palate shape is round in humans and it's rectangular in African apes. Also make special note of the words we are using here. Dentition, humans have no gaps between their teeth and they have smaller canines, whereas African apes have large canines and large gaps between their teeth, or gaps between their teeth, which are called diastema. Then we have humans, once again, they are non-prognathous. So that's looking at this part, this like snout region, the nose, and this protrusion in the front. That is prognathous. In African apes, they are prognathous. They do have this protrusion, whereas in humans, they are non-prognathous. And then we have the cranial brow. The cranial ridge is a bump, right, that is on the cranium, and African apes have that ridge, and they also have well-developed brow ridges. Those are these over here, whereas in humans, the cranial ridge is absent and the brow ridges are absent. So you need to study the diagrams of these, and here we have the pictures over here just to show you. They can give you the skull of the human and the skull of a chimpanzee, and then a gorilla, and you must be able to compare them. The same with the pelvis, the different pelvis. And then here you can also see the C-shaped spine and the S-shaped spine. And it's important to understand that in the gorilla, the foramen magnum is in a more backward position to allow the C-shaped spine to enter at the foramen magnum, and then in a more backward position in a more forward position in the Homo sapien to allow that vertical entrance of the spinal cord. Right, we are not going to do this question. Then we move on to the different types of evidence. Okay, we have fossil evidence. The transitional species serves as fossil evidence. These are characteristics which are common to both the ancestor species and the descendant species that follow. We have genetic evidence, and specifically it's mitochondrial DNA. Why? Because mitochondrial DNA is simpler than nuclear DNA. It's used to study the ancestry of females, and the mutations take place faster in the mitochondrial DNA. So it's easier to use it for genetic evidence. And lastly, cultural evidence, for example, is tool making. The development and use of tools separated humans from other hominids. Okay, so as they developed and as they became more well developed, they are able to, the intelligence improves and increases, and they are able to start using tools, and the tools become more advanced. Right, these are the fossils that are found in South Africa. You need to know them. You need to know the species. You need to know their nicknames, the site, and the scientist who found them. Okay, very important. Taung Child, Mrs. Pless, Littlefoot, Karabo, Homo naledi, and the Homo sapien, right? You need to know where they were found. Okay, then the out of Africa hypothesis, as I said, straight out of the examination guideline, you need to know that the fossil evidence that was there, okay, this is what it is, the fossil evidence that was there, that served for the out of Africa hypothesis, and the second evidence was genetic evidence using the mitochondrial DNA. Right, that comes to the end of our revision lesson. However, there's a few things that I need to mention, and that you must not forget, that you need to take into the examination room. And that is, don't forget your ruler, don't forget your compass, your protractor, your pen, an extra pen in case that one dies out, an eraser, a pencil, and also remember, when you have your ruler, it must be a ruler that has numbers and segments, because you use that ruler to draw a graph. You use that ruler to work out the scale. For your graph, you use the ruler to read off graphs that are given to you, to read off your phylogenetic tree, to measure things. It's very important. Protractor, ruler, compass. Please have all of your stationery. Prepare well, study hard, do your best, and use this opportunity to really, really prove to yourself that you can do it. Good luck for the exams. Until next time.